Hi, so it's my great pleasure to introduce Evan. So Evan is a graduate student in my lab from the BMSP, the program, the biochemistry program. And it's a fifth year graduate student, although I think you should count every COVID year as at least 7. So I don't know how what does that make him, right, in terms of implant. So he's been working really on one core project, and his sort of dedication of getting it through has been really amazing. Really applying kind of cutting edge computational and theoretical approach from information theory and machine learning to really try to deepen our understanding of really what's going on inside cells. So with that, I'll let Evan talk. And we're really at a point in this project that we're trying to kind of wrap things up and write it up. So any feedback you guys have will be really appreciated. Take it away, Evan.
Thanks. Yeah, it's been a while since I've presented in front of everybody, so I'm really excited to have the opportunity to do so today, even if it is remote. Yeah, so I'll be presenting today on phenotypic consequences of gene expression variability. Basically what that means is trying to understand how cell behaviors and cell responses are affected by the variance that occurs at the gene expression level, even among cells that are apparently homogeneous. So yeah, so I'm like, I always think this is a funny way to start, right? Like I'm just a bunch of cells talking about another bunch of cells. And even though, I'm made up of 30 trillion cells that are all completely different in their function and behavior, at the genetic level, they're all identical, right? They all have almost exactly the same DNA, or basically the exact same DNA. And it's only, for the most part, expression differences that have emerged that cause the functional divergence. And so there's a lot of layers of regulation that controls how that same common shared gene set are differentially expressed among a population of cells. And one of the most amazing features of multicellular biology. So there's, at the transcriptional level, variance can emerge, but even beyond that, there's regulation that occurs based on the identity and abundances of the proteins that actually carry out the tasks of expressing a particular phenotype. So there's post-translational modifications. There's the structures of the RNA themselves that can affect how what the abundances of the proteins are. There's protein interactions. All of these different layers create very complex emergent phenotypes that you get from just even a common gene set for a particular phenotype. So you have this balance between the information that exists at the expression level and all of the intermediate states that make the readout kind of complicated. So to kind of abstract the process a little bit, there's like one way you can think about it is for a particular phenotype, there's a set of relevant genes. Maybe that set is the entire gene set. Maybe it's a subset of all the genes. And the properties of the network are in part controlled by the rates of production and rates of degradation of all the components in the structure. So signaling network is made up of a bunch of proteins, and the abundance, instantaneous abundance of those proteins depends on the transcription, right? The rate at which they're being manufactured, and then also the rate at which they're degraded. And so calcium signaling specifically is a very good model system to kind of study this process because it's very well studied. There's a lot of models that kind of explore how the behavior can change under different stimuli. And it's also a very complex network, right? So there's examples of every kind of complexity that you can see in calcium signaling. There's everything from crosstalk to circuitous information flow. So like the stimulus, for instance, our stimulus here is ATP being exposed, the cells get exposed to extracellular ATP, which has to get propagated through a complex signaling network. And that's going to cause calcium to be released from the endoplasmic reticulum through this G protein sensor, this GTP mediated mechanism, right? So these receptors at the surface level, the P2I receptors, they mediate protein interaction so that when they get exposed to ATP, they cause a signaling cascade that causes phospholipase C, PLC, to start converting metabolites in the cell like HIP2 into a bunch of intermediate metabolites like IP3 and DAG, which then bind to the receptors and trigger protein kinases or trigger calcium release from the endoplasmic reticulum to cause protein kinases and phosphatases to start interacting with a bunch of different other signaling pathways and trigger gene expression that can affect the network in the future and affect decisions that are made in the future. And so one thing that's interesting to study about this network is there's like a trade-off between robustness and flexibility. The network has to be robust enough to be able to withstand all sorts of different fluctuations in the environment and fluctuations in the specific abundances of individual components. And then there's also like general constraints like resource availability at any given moment, and cell state fluctuations like cell cycle. All this network has to be robust to all those different things, but it also has to be flexible enough that it can convey stimulus information, or it has to be able to tell the cell about the dose and give it information about what to do. So how does variability in the gene set that populates this complex signaling network affect the ability of this network to convey information about the stimulus? And so one way of abstracting that is like, if you imagine all the genes that are relevant there, you can kind of make an axis, and there's like a distribution of what the individual abundances of gene expression products are going to be. And that a position of a cell in that expression axis is going to have some influence on, quote unquote, its full signaling capability, right? So if you're to compress all the distribution of all signaling responses into a single axis, right? Like, how does the position in your expression axis influence where you are and the signaling axis for a population of cells? And what you can call that joint distribution, the position on both of these axes at the same time, or the total set of allowable states that you might observe in a population is like the cell state space, and how big, how much tolerance, how many different expression states map to the same signaling space and vice versa, is a question that's hard to answer, but worth quantifying, because it gives you a lot of information about the sensitivity of the phenotype to specific fluctuations, and how much does gene expression variability really matter for this particular phenotype. And so the one challenge about this is that it's not just a single axis, right? These things are both of these spaces are very high dimensional, and for calcium, it depends on potentially hundreds of genes to a varying degree. And the actual signaling output is also very high dimensional. It's not just a single number, it's a time series. So it's got a lot of values that have some complex correlation structure. And so, right, like there's also the added challenge of the fact that the expression is a different data type, right? The signaling is a continuous process, whereas expression is discrete mRNA counts. So developing that mapping is like mapping those two spaces to each other will tell you what the strength of the dependency is. It allows you to quantify how important the gene expression variability is for a particular population of cells is on the output, and you can also calculate how much information is being conveyed, right, to the network, how much the capacity of the channel to calculate that information, or to transmit and propagate that information, just regardless of how complex the information flow is. It's an important thing to be able to quantify. And so these collective interactions of all these individual components cooperatively generate this emergent phenotype. How do you measure how strong that dependency is between the emergent phenotype and any particular layer of the regulation is really the question I'm trying to answer. And so luckily the data collection has been done before. So Rob Foreman, previous grad student, now working at VizGen, is he developed or implemented in the lab a pipeline for doing live cell imaging as well as gene expression, and I'll show the data in a second. But what's really difficult to do is, or what has yet been unsolved, both of them are very, very difficult, and luckily Rob has completed the first challenge. Second challenge of analyzing this mixed high dimensional data is still still a problem in data analysis, and is being worked on. And I think that the development in machine learning and deep learning has done a lot of work on trying to analyze high dimensional data, especially when there's mixed modalities, multimodal measurements. And so like a lot of approaches to doing this mapping involve feature engineering, clustering, dimensionality reduction, which are very useful for many tasks, but they're not guaranteed to capture all of the useful information in both spaces. And that's because they don't explicitly or exhaustively search for a mapping between the two spaces. So I think any approach that wants to really try to measure how much useful information there is that exists in both spaces has to do that process, has to exhaustively search for a mapping between them to find that that optimal. And so this is what the raw data looks like. So this is always such an amazing thing to look at for me personally. Seeing this is just a small subset of the cells. Total, there's over 5,000 cells. And this is just one frame, one field of view. And you can see that the responses are amazingly different, even among cells that are supposed to be genetically identical. They are homogeneous to every way that you can make sure that they're similar. The gene expression, this is the gene expression that you get from merfish. We have 83 calcium signaling related genes here. And so the way that I'm trying to solve challenge 2 is using information theory approach called mutual information, which tries to measure tries to estimate these probability distributions in calculated dependency. It's similar to correlation, but it accounts for nonlinear relationships too. And so I'll explain the approach for solving this in just a minute. But it's a non-parametric approach it's a parametric approach. It's not a non-parametric approach, which has a lot of advantages because the non-parametric approaches like binning and kernel density estimation are not really scalable to high dimension, and they also don't have you can't call them general purpose because they also don't scale well with sample size. And there's also other approaches that involve like making assumptions about the data distribution. So a mutual information doesn't need to make assumptions, or there's ways of calculating mutual information that doesn't need to make assumptions about the distribution of the data. So another way of looking at the raw data here, just to start about calcium raw data and understanding what is the total information in the calcium. This is what the signals look like for a small sample, and this is the diagram of the like showing all the signals all at once across seconds. It's 13 minutes roughly captured here. And the amplitudes, you can see that there's a lot of variability in the responses in the peak amplitudes and also in like the spikiness. And quantifying that's kind of difficult. And there's been a few approaches, and I don't think I've seen anybody use this particular approach. So FFT spectral entropy tries to estimate the frequency information in the signals. And just to test to make sure that FFT spectral entropy is a robust measure, not just calculating something that's sensitive to this particular scale or the particular dimensionality. So the way I tested that is by using just linear interpolation to change the dimensionality. You can see that some of the fine grain stuff gets lost when you shrink the dimensionality, and increasing the dimensionality doesn't change the amount of information, but it does if the metric is sensitive to higher dimensions, then it would output potentially more information just because there's more numbers in each signal. Then you calculate a periodicogram, which just shows the distribution of frequencies in the data and their relative amplitudes. And then you can calculate the entropy of that distribution. So the entropy of the signal distribution of the frequency domain. And this is actually very robust to affine transformations as well, like multiplying all the signals by a constant or adding, subtracting, etc. And so I don't show the changing scale and stuff, but take my word for it. I've tested them. It gives the exact same number regardless if you add, subtract, multiply. But what's very cool is that if you can change the calcium signaling dimensionalities. This is the periodogram that we use to calculate the signal distribution, the entropy of the signal distribution. You can see that it doesn't matter how large I interpolate out, as you'd expect, and you can even shrink the dimensionality pretty significantly before you see a significant drop off. And so this kind of represents like an upper bound on how much information there is in the calcium signaling. And it's useful to compare with the mutual information I calculate later, because the mutual information can never be larger than the amount of entropy in the smallest data distribution between the two data types. So if we have some reason to expect that 4.2 is kind of an upper bound there. It shouldn't be larger than this, assuming that the gene expression has more information. Now, so looking at the genes, you can also look at the correlation structure, try to understand how the genes are organized and how much information there is there. You can see that the gene expression is pretty, like pairwise, each gene is pretty weakly correlated to each other. Cell cycle genes, the handful of cell cycle genes that we have, they're much more likely to be correlated. But overall, the correlation between genes is pretty low. And so that has a question of, right, is it that these genes are all doing completely different things, or is it just random variance between them? What accounts for the fact that the correlation between genes is apparently pretty low? One way of answering that, or trying to answer that question, is to look at differential entropy and PCA. So PCA shows that you can explain most of the variance just with the first 6 principal components. But this leaves out part of the story, which is, like useful information. How much useful information are you losing in the rest of these, 80 or 70, 77 components, right? And calculating differential entropy, which doesn't tell us the magnitude of the entropy, but it does show us where the entropy is located across the principal components. You can see that actually most of the information is lost if you were to only take the first 6. Right, so the first 6 PCs explain 75% of the variance, but only 15% of the entropy. And it doesn't tell us how much of that entropy is actually useful for explaining the phenotype. Like that maybe it's useful, maybe it's not. How much, right? So 15% of the entropy, just a percentage is not super valuable. It's much more meaningful when you can say, this is the information that's useful, and this is how much extra extraneous stuff that doesn't really matter, just useless noise we can exclude by simplifying the process, seeing just how many genes really matter. Okay, so now I'm not going to have time to go super in depth on this, just going to briefly explain the process. So we use this mutual information neural estimator, MINE. It's a relatively new approach invented in 2018 for estimating the mutual information between high dimensional paired data, which is exactly what we have. It's really at the cutting edge of deep learning, trying to understand what is the dependency between high dimensional data types. And it does this by looking at that joint distribution, which I showed earlier as that like cell state diagram, and a permuted distribution, which is if you were to just permute the cells, right? So like disrupt the pairing that we have. We have one cell has this calcium and this gene expression. If you were to just mix up the gene expression axis so that one cell's calcium now is just paired with a random cell's gene expression, yeah, you can compare the difference between those two distributions is the mutual information. And it uses this Donsker-Varidan representation of the KL divergence to calculate the mutual information, or to estimate a lower bound for the mutual information. And it does this by exhaustively searching through the space of all mappings. So it tries to map the data distribution to the real domain. And calculating the supremum over this magic function here gives you a lower bound on the mutual information between the two variables, between P and Q. So this is the performance on the real data. This is what it actually looks like. So the mutual information between genes and calcium calculated to be 2.5 bits plus or minus 4, which is about 60% of the signaling information, that 4.2 bits that we estimate from spectral entropy, which is pretty significant. So that means that 60% of the information that's signaling, and that's in those signals, and all the signals exists in the gene expression variability already, and it's propagated through arbitrary layers, regardless of the structure and of the information flow in this complex network. And here I'm just comparing if you were to permute the data before feeding it into MINE versus giving MINE the correct pairing, what do you see? And the answer is very clear. After 85,000, 90,000 iterations, the information in the permuted sample goes to 0 over the average of, this is like six different runs, goes to 0, whereas the joint data quickly converges to the 2.5 bits that I'm showing here. Another way we can check this is to just compare pairwise for every gene versus like the mutual information that we get for genes versus the correlations that you get between genes and calcium. And looking at the data itself, I can tell you that there's not too many complex nonlinear relationships between genes themselves. So we expect these two heat maps to look very similar, and they do. They have like these general structures. The sorting is the same if you were to like sort by the average strength. This is just the cell cycle genes, but if you look at all the genes, it's also very similar. And just showing this for sake of simplicity. The advantage of using the mutual information is that if there was nonlinear relationships, it would be captured, and it also measures the true dependence in a way that we can then compare these numbers to the numbers we get from the full data. So the bits that we get in each one of these boxes is truly comparable to the number we get of bits between both the full data distribution. So it's much more useful for comparing, and in the higher dimensional spaces, the nonlinear relationships are much more important. Okay, so moving along quick. So this is the mutual information between every gene, all 83 genes, or the 83 genes that we kept in the data that we measured with Murfish, and the calcium signal, the full calcium signaling. So seeing how much genes inform about calcium signals. You can see that there's quite a big range, right? Everything from like 0.1 bits or even non-informative to 1.5 bits, which is pretty close to the full amount, right? So 1.5 bits is like over 70% of the full information between the two. Which begs the question, right? Like, what information are these genes containing? They must contain information that is stored in other genes too, right? Like this is clearly over accounting. And if you like sum up all the information in every one of these genes, you get like 56 bits as opposed to the 2.5 we get for the full gene group. So it's clear that there's overlaps and complex relationships between groups of genes, not just individual genes that explain why the full number is so much smaller than the sum of the individual. Okay, what accounts for all these complex relationships? And also just analyzing individual genes, right? We can like CCDC47 is calmodulin dependent, PPP3CA and 1CA. Those are both phosphatases. And the relevance of them being phosphatases is like this crosstalk that I mentioned earlier, right? Like phosphatases and kinases are some of the most broadly interconnected proteins and signaling networks. So if you go through an account, you'll find that the top 10, top 20 is highly enriched for phosphatases and kinases, right? These general proteins that interact with many others. So it's interesting that the most interconnected proteins tend to be in the ones that are interacting with a lot of regulators tend to contain more information about the phenotype. And this is just showing that sum, right? If you were to do a cumulative sum of the number of genes, it's clear that the individual genes inform way too much. It's like 10x the signal entropy, and the average gene is 0.7 bits. So how do you explain the groupwise interactions? So the first way we explored that was looking at pairwise interactions with calcium. So not just one gene with another gene, but pairs of genes with calcium. Oh, sorry, this diagram right here, what I'm showing is one gene versus another gene. And the one gene versus another gene, you can see that genes generally aren't super informative about each other in absolute magnitude. Like the average mutual information between one gene and another gene is 0.04 bits, which is pretty tiny compared to what we see, comparing to that diagram I just showed, where each gene has on average 0.7 bits about calcium. But it is interesting that you can correlate, like the correlation between the amount of information one gene has about another versus the amount of information that gene has about calcium. There's a positive correlation. So genes that are more informative about each other, about other genes, are also more informative about calcium. So it seems like you might be able to abstract this and say like, okay, there might be some summary genes, right? Like genes that are very informative might contain summary information of those others, but do they have any unique information about calcium? And so one way of breaking down this like synergy versus redundancy type argument, like how much information do genes contain at the group level that they don't contain at the individual level versus how much information do they share, right? There's this tool called SRI, Synergy Redundancy Index, that measures the distinct information that exists in pairs of genes. So what we calculated is the mutual information of between a pair of genes and the full calcium signal, and then subtracting out the information that one gene has about calcium and the other gene has about calcium. So if this value is positive, then it means that the sum of these two is less than the value of the combination, which means that there had to be extra information in the pairing that didn't exist in either alone. If this value is close to 0, then it means that the sum of these two is about equal to the pair. So the sum of the parts is equal to the whole. And they're providing relatively distinct, unique information. If the value is negative, it means that the sum of each individual gene is greater than the pairing, which means that the pair had redundant information with each other. These two genes have redundant information. information overlap. So this is the full SRI matrix between all pairs of genes and calcium. And it shows that there's mostly redundant information. I'll show the histogram in a second so you can see just how redundant it is. What is interesting is PPP3CA and CCDC47, those are the most informative genes that we saw individually. They're also the most redundant. And what you'll find is, and I'll show in a second, that the more informative a gene was on that first figure I showed, the more redundant information they had on average. But it's also interesting that genes that appeared completely uninformative individually on that graph before also became more informative in a group, right? Which is why they have positive synergy. So the synergistic genes, it's not that these genes are maybe uninformative as the previous graph that I showed, like histogram that I showed. Not that they're uninformative, it's that they might provide some contextual information, or the sample size is too low to reveal the information. But when you see them in a group, suddenly there's this like emergent information that the gene they're paired with didn't have. So even informative genes, right? Even genes that were otherwise informative have these positive values when paired with what appeared like uninformative genes. This is just the histogram in that correlation. The important thing here is that if you take the mean row of the SRI, mean row, and you look at the mutual information between a particular gene and calcium, you see there's a negative correlation. The more informative that gene was about calcium, the more redundant it was on average with all the other genes in the okay, so moving right along. Now this is the kind of final couple slides looking at how at the group scale. I think this is where the most interesting thing is. So what we did is we wrote these couple of approaches for searching through the full group space. There's a greedy algorithm, a minimal algorithm, and kind of just random. So the greedy algorithm looks for the most informative gene and adds it to the set. And when the set has one, then it goes through all the remaining genes. So there's n minus one, right? 2 genes after the first gene is added, and it looks to see which of those genes in combination with the current set adds the most information. And then it keeps doing that until it's reached all 83. So it's basically a limited way or a constrained way of searching for the most informative set of genes and building that as you go. And the minimum algorithm does that, but the exact opposite. Instead of looking for the most informative gene to add to the set, it looks for the least informative gene to add to the set. It's always calculating the information between the current set of genes plus the possible extra to add and in calcium. And if it looks for the addition that added the least amount of information. So looking at this graph, I think the one interesting thing to do is look at the shape of this minimum distribution is pretty linear. And the fact that it's almost always increasing indicates that there's always unique information that you can add, right? Like every gene that you add to the set adds information. So you calculate the slope of that, and that's 0.03 bits. Tells you on average, every gene that I can't find a gene that adds less than 0.03 bits to the set. They all have some unique information that I can add. Another thing, looking at the green line and comparing the green line to the purple line, PPP3CA is kind of the star of the show a little bit. Always adds unique information. It has the most information total, so it was the last one to add to this purple set. It was also the first one to add to the green set. So not only does it have the most information, but it also has the most unique information, which I think is kind of interesting. It's not just that more genes contain summary information, it also contains the most unique information. And then looking at this graph is kind of showing a comparison of, like, if you were to do that total, right? So this first line in the set is that 56 bits that you get from adding up all the individual genes, how much information they contain about calcium, and then adding groups. What's the expectation of how much information that all possible groups of a given size would have about calcium? See that the line drops off pretty quick, but it's not that the genes are fully redundant. So this would be if every group had the same amount of information. Instead, you can see that it really quickly diverges at 6 genes, and the amount of unique, like synergistic information that these genes have diverges pretty quickly. Okay, so last data slide, wrapping up, just comparing, kind of bringing it back to the beginning. We look at this like differential entropy graph versus the actual information between all those principal components in calcium. you can see that every principal component adds information. It's not like it grows exponentially. The amount of useful information appears to be a lot more linear than this would suggest. So you, by using principal components, cutting off the principal components, you are losing a lot of irrelevant information, but also as you explain more variance, you're just always adding more information. So it tells you that there is a lot of unique phenotypic information even in that last principal component, even though there is like maybe a diminishing return in terms of the number of principal components. But it does suggest maybe if you added a lot more genes to this, maybe the trend would continue. So mapping dependency, this is kind of a really useful case study, just mapping the dependency between transcription and phenotype. Like this is a number like 60% dependency. I don't think anybody could have told you prior to the application of this method. But the real power of this here, I think, is that you can do this between any two layers of regulation, anything that you have paired data for with a sufficient sample size. It doesn't matter how many layers of regulation there are in between or what the complexity is of the functional relationship or the circuit that propagates the information from node A to node B. And with that, I'd like to thank everybody. And we don't have a new group photo to take in because of COVID, but seeing this is kind of a useful reminder of how big we used to be and how all the cool things people have done, in the time I've been here. So I'd like to thank especially Rob for creating the data and being very useful, and Roy for all of his guidance, teaching me basically everything I know about statistics, and everybody else in the lab. So thank you very much.